First, of course, is that the selection process is slightly elaborate, and it is a two-step selection. You have to plate the cells twice to actually get to the final recombinant transformants, and that too in indirect manner. A one-step color-based assay for differentiating recombinant transform cells from the non-recombinant ones would be more convenient. So to address this limitation, we moved on to Packet and Packet Team, which can basically give you a color-based selection, and this process is known as blue-white screening. We'll see this in details as we move along, and we'll also go into the molecular basis of blue-white screening. Then, of course, the second important limitation with almost all plasmids is the maximum insert size that they can hold. So not more than 5 to 6 kb of insert can be cloned into PBR322 or other plasmids, and that is a major concern. So the enzyme that plays a key role in blue-white selection is beta-galacticidase, and beta-galacticidase is made up of two distinct functional parts, the omega fragment and the alpha fragment. So here is a cartoon of beta-galacticidase, and there are 4 copies of omega fragment and 4 copies of alpha fragment, and the omega fragments are polymerized in the presence of alpha fragment, and that allows for the synthesis of a functional beta-galacticidase. Right? Beta-galacticidase is an enzyme that is encoded by the host, and this gene is a part of the 2 other genes found in the lactoferrons. So I'm sure you would have a basic idea of the functioning of the lactoferron, and lactoferron basically consists of 3 genes, and of the 3 genes, beta-galacticidase is an important gene because this is the one that controls the conversion of lactose to glucose and galactose in the absence of glucose. So when the glucose is not present in medium, then as an energy source, lactose is used and it is metabolized into glucose and galactose. These are inducible operons, that is, upon addition of a substance, specific substances, these operons can be induced to express. And specifically in conditions of less glucose, the lactoferron is expressed. And when the lactoferron is expressed, beta-galacticidase is one of the enzymes that is synthesized. Beta-galacticidase converts lactose to glucose and galactose. So the innovation that is done to introduce blue-white screening in plasmid vectors is the fact that the host cell lacks the gene is mutated so that it can only produce the omega fragment and not the functional alpha fragment, right? So therefore, as long as the host cell does not have a plasmid that can actually allow for coding of the alpha fragment, the host cell will not be able to synthesize a functional beta-galacticidase. The absence of alpha fragment is complemented through the plasmid. So the alpha fragment can only be synthesized in trans by the non-recombinant plasmid. And even in the plasmid, you have innovation in the sense that the multiple cloning site of the plasmid is introduced in between the alpha fragment coding region. So here is how a typical path vector is formed. Path vectors are derived from PBR322 by modifications. There is no tetracycline resistance gene here. You have only one resistance marker, that is the ampicillin resistance marker. You have the equal origin of replication to ensure that the plasmid can autonomously replicate. The tetracycline resistance gene is gone. We have the addition of LAGZ prime alpha, which is the locus from where the alpha fragment of beta-galacticidase is synthesized. Also, what is done is that the MCS or multiple cloning site is within the LAGZ prime alpha locus. So now when you have a recombinant plasmid, the LAGZ prime alpha locus is disrupted, and therefore this cannot synthesize a alpha fragment. However, in a self-ligated plasmid, the LAGZ prime alpha locus is intact, and therefore this can synthesize the alpha fragment, which can then combine with the omega fragment from the host and give you a functional beta-galacticidase. So that's the whole strategy behind blue-white screening. Path vectors are derived from PBR322. Only the ampicillin resistance gene is present, and there is no restriction enzyme within the ampicillin resistance gene. The tetracycline gene is replaced by the LAGZ prime locus. This codes for the alpha fragment of beta-galacticidase. And very innovatively, the MCS is within the LAGZ prime, and the MCS has been expanded to contain a lot more restriction sites than are present in PBR322. And the selection process is based on the LAGZ prime expression. And as I said, a functional beta-galacticidase is formed only when the LAGZ prime locus on the plasmid is intact, which essentially implies a self-ligated plasmid. And this is assayed by the blue color of the colonies in the presence of XGal and induction of lactoferron by IPTG in the host cells. So the process by which a defective beta-galactosidase gene in the host cell is complemented by the expression of the alpha fragment through the plasmid and trans is referred to as alpha complementation. And PAC vectors are high copy number plasmids. Each cell can contain as much as 500 to 700 PAC plasmids. So let's see what happens during the synthesis of beta-galactosidase in a mutated host cell. You have your mutated LAGZ locus in the host cell that synthesizes only the omega fragment and is not able to synthesize a functional alpha fragment here. So therefore, this omega fragment dimers are inactive and cannot perform the function of beta-galactosidase. However, what you can do is, if you could provide the alpha fragment that is synthesized through the sequence in the plasmid, then of course, you could have the formation of a functional beta-galactosidase, and this could then perform the function of breaking down lactose to glucose and galactose, providing the necessary energy for the cell. Right? So in this case, the omega fragment is coming from the host cell. The alpha fragment can come from the plasmid, and this is again very important that the plasmid that can synthesize alpha fragment would be the one that does not have the insert or the non-recombinant plasmid, because the MCS or the multiple cloning site has been very intelligently placed inside the LAGZ prime locus. LAGZ prime locus is the one that codes for the alpha fragment. So when the insert is present, the LAGZ prime locus is disrupted, and therefore no synthesis can happen. However, if it is a self-ligated plasmid, then the alpha fragment can be encoded in trans via the plasmid here. Active beta-galactosidase is a tetramer. As is shown here, there are 4 omega fragments and 4 alpha fragments that join together to form the functional beta-galactosidase. Right? Each monomer is made up of 2 parts, the LAGZ alpha and LAGZ omega. So in the non-recombinant transformants, the omega part comes from the host cell, the alpha part comes from the plasmid, and the 2 combine together to give you a functional beta-galactosidase. Right? The M15 deletion in LAGZ gene in the host cell deletes the alpha fragment. So the omega fragment cannot tetramerize by itself, and therefore you do not have a functional beta-galactosidase. However, the functionality can be restored by providing the alpha fragment in trans via the plasmid here. So this is your alpha fragment coming from the non-recombinant plasmid and forming a functional beta-galactosidase in combination with the omega fragments here. And this process is known as the alpha complementation process. Let's look at how blue-white selection allows you to discriminate between non-recombinant transformants versus recombinant transformants. So here is what is happening. The host cell is mutated, and therefore it cannot synthesize the alpha fragment of beta-galactosidase. It can only synthesize the omega fragment. And the problem is that the omega fragment cannot itself polymerize and give you a functional beta-galactosidase. So therefore, what we need for the polymerization of omega fragments is the alpha fragment. In case of non-recombinant transformants, this alpha fragment is provided by the LAGZ prime locus in the plasmid, which is also holding the MCS. So here in this case, what happens is that the non-recombinant plasmid would have an entire LAGZ prime locus, which can synthesize alpha fragment, and this alpha fragment can then combine with the omega fragment derived from the host cell, forming a functional beta-galactosidase. Right? So this is what happens in case of the non-recombinant transformants, which have a self-ligated plasmid where the LAGZ prime locus is intact, and therefore the alpha fragment can be synthesized, and these alpha fragment then combine with the omega fragment to form the beta-galactosidase. However, in recombinant transformants, because the MCS is within the LAGZ prime locus, the LAGZ prime locus is itself disrupted by the insert, and therefore no alpha fragment synthesis takes place here. Effectively, there is no functional beta-galactosidase because the omega fragments cannot polymerize by themselves. So therefore, there is no beta-galactosidase function found in the recombinant transformants. Right? Now, how do you discriminate between the recombinants and non-recombinants visually? For that, you use what is known as XGal, and XGal is converted into a blue colored product upon the presence of beta-galacticidase. So here you are, this is your XGal. So XGal in the presence of IPTG is the inducer for the lacoperon. Once the lacoperon is induced, you have the formation of beta-galacticidase, in case of non-recombinant transformants, resulting in development of blue colonies here. So blue colonies are because of the conversion of XGal to blue color by beta-galacticidase. In the case of recombinant transformants, there is no functional beta-galacticidase, and therefore XGal is not converted to its blue colored product, and therefore you have these white colonies versus blue colonies. Blue colonies are the non-recombinant colonies, white colonies are the recombinant colonies. So this is typically a blue-white selection. You have the spectral plate which has been plated with your transformants, and you can see distinctly there are some colonies that are blue in color, and then there are colonies that are white in color. The blue colonies represent your non-recombinants. The white colonies are the ones that represent your recombinant. So in the blue colonies, XGal is converted to its blue-colored product. While in white colonies, because beta-galacticidase is not present because of the lack of the synthesis of alpha fragment, you have only white color and no conversion of XGal to blue color. So white colonies, these are colonies with recombinant plasmids. Lag Z prime is disrupted in the plasmid, and therefore functional beta-galacticidase is not formed here. And then you have the blue colonies. The blue colonies are the colonies with self-ligated plasmid, where the lag Z prime locus is undisrupted. Therefore, a functional beta-galacticidase is formed upon the combination of the omega fragment from the host and the alpha fragment from the plasmid. This acts on XGal to give blue color product upon induction by IPTG, right? And these are your full forms. The XGal stands for 5-bromo-4-chloro-3-indolel beta D galactopyranoside. IPTG stands for isopropyl thiogalactoside. Packetine vectors can work with modified host cells where there is a deletion in the lag Z gene, so as to ensure that the alpha fragment is not synthesized from the host cell and only the omega fragment is synthesized from the host cell. And therefore, a functional beta-galacticidase cannot be formed by the host cell alone. The alpha subunit is encoded by the lag Z prime locus in the plasmid. So here, if you see, this is the lag Z prime locus in the plasmid, and very interestingly, the MCS or multiple cloning site, which is a series of recognition sequences for restriction enzymes, is present within the lag Z prime locus. So if you have your insert ligated onto this locus here, then your lag Z prime locus is disrupted, and therefore a recombinant plasmid cannot synthesize the alpha fragment, whereas a self-ligated plasmid can synthesize the alpha fragment here. So importantly, the lag Z prime locus also contains the MCS, and the placing of MCS within the lag Z prime locus allows you to control the synthesis of alpha fragment of the beta-galacticidase. In recombinants, no alpha fragment is synthesized. In non-recombinants, the alpha fragment can be synthesized and therefore leads to the formation of a functional beta-galacticidase. Thus, in recombinants, the alpha polypeptide is not synthesized. Hence, no functional beta-galacticidase is formed here. In non-recombinants, however, the lag Z prime is intact. The lac operon is induced by IPTG and XGal and rendered blue by the functional beta-galacticidase that is synthesized in non-recombinant transformants, right? Thus, when the cells are grown in ampicillin plates with IPTG and XGal, the non-recombinants turn up as blue in color because of the synthesis of functional beta-galacticidase, and the recombinants turn up as white in color because the XGal is not converted to blue-colored product in the absence of a functional beta-galacticidase. So while blue-white screening solved the purpose of doing a one-step selection for plasmids, the other major limitation of plasmids remained, that is the maximum size of the insert that you can have in a plasmid is around 5 kb. You can restrict it to a max of 6 kb, but not anymore. And many a times your insert may be of a larger size depending on the experimental objective that you want to achieve, for example, in sequencing. So one of the major limitations of the plasmid vectors, that is two-step selection process, was removed using blue-white screening. However, another major limitation with respect to the size of the insert that it can hold remains. So not more than 5 to 6 kb of the insert can be held by a plasmid, and therefore the search for alternate vectors began, and this yielded several alternate vectors, so starting with bacteriophages, which we'll discuss in the next lecture now, right? But before we close this one here, I want you to again do an activity and enumerate for me the role of the following in blue-white screening: beta-galacticidase, XGal, and IPTG. So on that note, we'll close here, and I'll see you again in the next class, and we start with lambda fathers as vectors. Thank you, class.